Good morning, everyone. If it's okay, I'll just stay down here. I came to FDA from academia, so I'm more comfortable being closer to the audience. So thank you again. Let me grab this. So standard disclaimer that everyone's had. So the objectives for today's talk will be first to look at some of the key regulations that govern clinical pharmacology-related information and labeling, including our recent guidance that was published almost a year ago. Second thing will be we'll look at some alternative display methods to help with the readability, clarity, and utility of labeling. And then last, we'll discuss some strategies for some unique situations that you might find in the clinical pharmacology section, such as modeling and simulation related data and in vitro data. So you've seen these regulations several times yesterday, so I'm not going to go through them point by point, but rather I'd like to put them into the context of clinical pharmacology related information. So what do I usually see when I see a clinical pharmacology label come in to our office? Well, in many cases, what we'll see is it's really sort of a, I don't want to use the term data dump, but very close to what we see in the executive summary from the clinical pharmacology summary in the document. So it's very complex information, very technical language, a lot of clinical pharmacology jargon, and I love it because I'm a clinical pharmacologist, but when I put my clinical hat on, or I talk to some of my clinical colleagues who came to us more recently from practice, they find it very challenging. So we have to keep in mind that the label is written for the healthcare provider. And we really have to think about, and I tell this to reviewers as well, is what information do I need as a regulatory officer to make regulatory decisions versus what information does a healthcare provider need to make prescribing decisions. And it's not always the depth of information that we have in our clinical pharmacology summary in the NDA package. So we really need to think about the essential information that we need to put in the clinical pharmacology section of the label, and do we need to word it in a way that it will be understandable to someone who's not a clinical pharmacologist? Those are two really key points. Information needs to be updated. A problem with, in particular with clinical pharmacology, drug interactions are always changing. We're finding new transporters, new metabolic pathways, and that's something that we always need to worry about is this section up to date, especially with those things. Promotional tone or being false or misleading. Sometimes the clinical pharmacology section can lend itself to promotional ways of presenting information because sometimes we talk about, for example, linearity or exposure response where we're talking about dosing ranges or different biomarkers and stuff. And depending on how you choose to word it, it can sound very promotional. It can promote an unapproved indication or it can promote an unapproved dose. So we need to be cognizant of the fact that that's not really the spirit of the regulations, and actually it's against the regulations to be promotional. So we need to be cognizant that that could be happening. Lastly, clinical pharmacology is a translational science, which means we tend to translate preclinical information and clinical information and try to meld them together. So we do have a lot of non-human data. So there's a lot of in vitro information, there's a lot of animal information, and we have to be careful about what information we're putting in labeling. Is there human data that superseded the in vitro data, and therefore does it need to be in the label? Probably not. Animal data hasn't been superseded by human data, probably doesn't need to be in the label. So this slide just shows the breadth of clinical pharmacology's footprint in the labeling, which is probably greater than 50%. The bulk of the detailed clinical pharmacology information you'll find in section 12, which is the bottom of the pyramid there. However, that informs many of the actionable sections that you heard about yesterday. So it would inform specific populations, drug interactions, dosing and administration, some of the risk-related sections. And really the way it works is, since it's a higher numbered section, there you would find really the details of studies and it informs the actions that you would find in the lower numbered sections. So like I said, about a year ago, we published the final guidance for Section 12, the clinical pharmacology section of prescription drug labeling. And really that addressed three big areas. So the what, the where, and the how of putting information into the section. So what, the content, where. So the what, we didn't have too many changes. There's some new information about modeling and simulation data. The where, there were quite a few changes. We'll talk about them, formatting, putting things slightly in different places and whatnot. And then really the how. This was kind of a new thing where we really focused based on CDER's labeling initiative and our own OCP labeling initiative that we wanted to let industry know that we were really looking at communicating, optimizing communication of this information. That is, making it more readable, more clear, more useful. And we gave options for not just using text, but using tables or figures where appropriate. So all that was in the new guidance. So first, let's look at the structure of the section. So we have the section heading, section 12. Then we have three main subsections: mechanism of action, pharmacodynamics, and pharmacokinetics. Then we have two other designated subsections: microbiology and pharmacogenomics. And those two you would use only if you had data that to put in there. So if you didn't have any microbiology information, if it's not that type of agent, you wouldn't have that. But those are designated numbers. So if, for example, you wanted to put an additional subsection, you wouldn't, and you only had 12.1, 12.2, 12.3, you would have to start the additional subsection at 12.6 because those other two have already been designated. And when would you use an additional section? You'd use an additional section if you had information that clearly didn't fit under any of the other subsections, or if you had information of such great breadth or complexity that putting it into one of those other headings or subsections would make the other information, unable to read because it would be so, voluminous. Other things, so in the pharmacokinetics subsection, we've restructured the headings and subheadings to make it a little bit more intuitive. So rather than having ADME, absorption, distribution, metabolism, excretion, we've changed it to absorption, distribution, elimination, and under elimination we would put things like half-life and clearance that don't really fit under metabolism or excretion, and then we'd have metabolism and excretion section. We also changed the wording of some of the subheadings under specific populations to make them more consistent with the information that's underneath them. So the next 4 slides are just some general concepts I took out of the guidance. Obviously, I would suggest you read the guidance itself so you understand all this information with the appropriate context around it. These are just, like I say, general do's and don'ts. There's obviously going to be times when things don't, there'll be exceptions, and those we can deal with on a case-by-case basis, but these are just some general things to think about. So first, looking at the content and organization of Section 12. Of course, one is it has to be understandable to the non-clinical pharmacologist healthcare provider. Another big issue with regard to content and organization is consistency. So we'd like to see consistency in how you format your headings and subheadings. It should be consistent with how it's done throughout the PI. So if you use italics for headings and underline for subheadings, it should be that way throughout the document. You should try to be consistent in your units and parameters that you use. So for example, you shouldn't have molar and then switch to nanograms per ml, then switch to milligrams per ml. Because while that might be very important to me as a clinical pharmacologist, a healthcare provider may not, when looking through this quickly, understand these transitions. And if it is clinically important to have those transitions, then maybe we need to explain what that means or why those need to be that way. Another issue, again, it deals with the promotional misleading, which we talked about. So we shouldn't use promotional and misleading language. We shouldn't try to suggest any or imply any unapproved indications. We need to be careful with subjective wording. So words like fast, rapid, selected, selective. I worked in 7 years in DDMAC, which is now OPDP, so I know all these words. They jump out at me when I see them in labeling. So we should try to avoid doing that. Or if those words go in there, they need to be quantified. If you use general terms, such as exposure, if I were to ask several pharmacologists what the word exposure means, one might tell me it means area under the curve, another one would say area under the curve and Cmax, another one would say area under the curve, Cmax, Cmin, C average. So it means different things to different people. So if you choose to use a general term like that, we should define it at least once in the document. Lastly, just with regard to animal data, again, if we have clinical data, we should really not use in vitro or animal data that's been superseded by clinical data. The pharmacodynamics subsection, which would be 12.2. So the big things that were covered in there were first, what should go in there? So it should be PD effects, information about onset, reversibility. It should include the important PD biomarkers that may have been identified. It should include exposure response, exposure safety relationships. An important reminder we put in the guidance is that there is a regulation that says if there isn't information on these parameters, that we need to put a statement in there saying that the exposure response is unknown or something to that effect. A new thing for this subsection is that if you have drug interactions or patient characteristics, specific population type information, that's only PD related. So for example, you have one drug that causes bleeding risk, and you have your drug that also can cause bleeding risk, and together it's a 1 + 1 = 3 situation where you have mild bleeding, severe bleeding. That could and it's just a pharmacodynamic effect, and that would go into 12.2. If PK is driving the PD effect, so we're having two drugs together, increased concentrations causing increased bleeding, then that would go into 12.3, the pharmacokinetic section. So if you have pure PD drug interactions or pure PD related patient characteristic effects, age, whatever, that would go into section 12.2. With regard to the heading for QT studies, so if there is a QT study done and there's no QT effect, we have standard wording that you could use to express that. So it's really just pretty much one line. If the QT effect is unknown, you don't need to put a heading and then just say unknown. You could just leave the heading out. So that was described in the guidance as well. With regard to presenting, especially the exposure-response relationships, again, we have to be careful about promotional tone. I mean, sometimes it appears that folks try to use this section maybe perhaps as a backdoor to get stuff in, clinical outcomes or whatnot that probably wouldn't get into 14, but you'd really like to get into the label. So that kind of thing is really not, again, in the spirit of this section, and we should try to avoid doing that type of thing. Okay, moving to the pharmacokinetic section. One big change is now we have what we call a general introduction section, and it goes from the subsection title pharmacokinetics to the first heading, which is usually absorption, assuming it's an oral product. And in there you put a lot of the information that won't fit into the other headings and subheadings. So in there, for example, you'd put information about linearity. You'd put information about accumulation. You'd put information about, is this a prodrug? If the exposure numbers are important, the actual AUC or Cmax, you would put that information in that general section. And then you would have the other sections: absorption, distribution, elimination, metabolism, excretion. Under absorption, given that food effect studies have become a little bit more complex, so it's not just the old high-fat meal anymore. It could be high-fat, low-fat, medium-fat, calcium-rich, calcium-poor. Sometimes this information can become fairly complex. So because of that, we gave the option of putting in a subheading for food effects under the absorption heading. Under elimination, with regard to half-life, we generally recommend that the effective half-life be used. That would be the half-life that we use to define steady state. Sometimes drugs have a longer terminal half-life, and if it's clinically important to put that information in there, then obviously we should put it in there, but routinely we might not see it in there. If you have a drug that has nonlinear pharmacokinetics, that is, you increase the dose, let's say twofold, and you get a 30-fold increase in exposure, it's nonlinear, you need to give the half-life, if you report it, you also need to give the dose associated with that half-life because it's dependent on that. So that's something that's new. With regard to metabolites, we should only be including information on metabolites that contribute to the efficacy or potential toxicity of the drug. And if it's an active metabolite, then we should be putting the percentage, the relative to the parent, the percentage exposure that's the active metabolite, and we should also put the relative potency of the active metabolite to the parent compound. We shouldn't be putting information in regarding pathways that have been ruled out unless we don't know completely what the metabolic pathway is for the drug. So if we have very limited information, then we could say, well, we ruled these out, we still don't know how the drug is metabolized. So in those cases, you could put it into the label, but in general, we don't put pathways that have been ruled out in the label. With regard to the food effects section, as with most of the information in this section, we don't put or repeat information that we've put into other sections. So for example, you would give a description of the food effects study, perhaps, the results of the food effects study, but you wouldn't say the conclusion or the action, give with food, give without regard to meals, or whatever. That would go into section 2, so that wouldn't be repeated here. And that would be the same for drug interactions. It would be the same for specific populations. That information would go into the actionable sections, 7 or 8. So specific populations and DDI studies. So both of these, for specific populations, is patient characteristics that affect the exposure of the drug. So things like age, race, organ impairment, disease effects, other things like that. How that affects exposure. Drug interactions are obviously combining two drugs and having it have either some change in exposure or PD or something like that. And if it's purely PD, then it would be in 12.2. So those can be broken down further into either interactions or patient characteristics that result in what we call clinically significant effects and those that aren't clinically significant. So the ones that are clinically significant, we generally need to provide more details because we're giving context for the regulatory conclusion and actionable information we're putting in section 8 or 7. So in those situations for a clinically significant, either patient characteristic or drug interaction, we would put information about the results of the study, so the exposure change, for example. And we may put some information, brief brevity is definitely recommended, about the study itself. And that would depend on really the totality of information that we have. So for example, we may have information that suggests that the drug behaves differently in patients versus healthy volunteers. So in this case, it may be important to say that patients were used versus, or healthy volunteers were used in a certain drug interaction study. We may have information that, for example, in the drug interaction study, they used a lower dose of an inhibitor. And we may believe that, if they used a full dose, we'd see an even greater change in exposure. We might want to tell healthcare providers that you may see a higher exposure than reported here. You may want to put something like, or put the dose in. The thing to remember, though, is just putting those facts in. As a clinical pharmacologist, I'll, of course, pick up on the fact that, okay, the dose of ketoconazole is 200 instead of 400, or it's in healthy volunteers and there's an issue. But really, is the healthcare provider always going to pick up on that, especially if they're reading it quickly because someone's having an adverse reaction or something like that? So is it better to explain that information rather than just put in the fact that it was done in patients and assume the healthcare provider is going to figure out what that means? They might not. So I covered it all. Oh, this is the last thing I want to mention. Just regard to the PLLR. So lactation information no longer goes in the specific population section. That goes in section 8. So just keep that in mind. When you present information, this type of information, like I said, there's clinically significant and clinically and non-significant information. A lot of times, especially when you do it in figures, they'll mix the information, and it's very hard to tell what's clinically significant and what isn't. Yes, there's 90% confidence intervals, but there are times when the 90% confidence interval difference doesn't really relate to a clinically significant risk or something like that. So it has to be clear in how you if you present the information together, it has to be clear what's significant and what's not significant. And one way that we recommend doing that, and this is for 12.3 and 12.2, is that you don't need to provide a lot of detail for negative studies, non-clinically significant studies. So in most cases, you could just provide 1 or 2 sentences that said, the following drugs did not show clinically significant change in exposure, and drug A, B, C, D, E, something like that, or characteristic, race, age, whatever, and just do it that way. And then have the more detailed information, and you could use tables or figures for the drug interactions, and just have, like I say, the negative information or the non-clinically significant information just as a one sentence thing. So before I go to alternative displays, I want to say that so we've been almost a year now since the final guidance was published. And it's been very encouraging with regard to the formatting changes and some of the content changes that we recommended. And it's actually I'm very happy about that, because when I see stuff come in, I'm starting to see it formatted, consistent with what I was just talking about. The one place that I'm not really seeing much movement is in that how thing. They're really trying to bring the information down to the essential information, trying to communicate this information in a way that's not highly technical, trying to use alternative formats to sort of break up the information so it's a lot easier to read for someone who's trying to read this stuff quickly. So I presented these displays at least 4 or 5 times at clinical pharmacology meetings around the country, so various societies. And usually at the end, I'll get someone from the drug company who will come up and say, this is really interesting stuff. However, I'm a clinical pharmacologist. I don't really do labeling. I'll get a call once in a while. But you need to talk to the labeling people and tell them they need to do this. So here I am. Labeling people, please, let's communicate information better in this highly technical, highly complex section. We have to start somewhere, so why not start here? We're interested in doing it. Hopefully you guys are interested in doing it as well. Let's try to make this section better. I think we can do it. So with that, I'll move into some alternative displays. And these are just suggestions, ideas. They're not templates or required formats. So this first one is using a tabular format for the pharmacokinetic information, so the 12.2 information, for a fixed combination product. So normally with this type, you would have lots of text, one component, and the next component, and the next component, a lot of text and information. Healthcare provider trying to read it quickly is going to get lost. So this is perhaps an easier way to present all of that information concisely rather than have all that text. So that's one. And we've actually used this. We've had some approved products where you'll find this type of table in there. Another time would be for a single product where the information is very complex. So here we have a lot of complex information. We have 2 exposure levels. We have single dose and steady state. We have multiple arms of your food effects study, and we have a whole bunch of active metabolites that we have to worry about. So if we were to put this in text, it would be very complex, a lot of information. So putting it in a table is perhaps an easier way to present all of this complex information. It's easier for a healthcare provider to navigate to the information they're particularly looking for and include that information. Our specific populations, so again, these are the patient characteristics. This is presented in a figure format. It could also be done in a tabular format, which is the next slide I'm going to show you with a drug interaction example. But here you see those vertical lines. What those are boundaries for when we determine something to be clinically significant, where we're going to do something, avoid, change the dose, monitor more, do something. Where you have the ability to do that, I think it's a good idea, my personal opinion, because it really gives healthcare providers an idea of where these actions are coming from in the other sections. So it's very helpful when we have those. Sometimes we don't have the ability to make those calls, but when we do, we should do that. So this is a tabular presentation of drug interaction information. Again, this information could also be done in a figure similar to the previous slide. So both of those could be either tables or figures. And in this case, you can see we have the clinically significant drugs here in the table, and then we have the non-clinically significant drugs in that one line down below. So it's very clear what's significant and what's not. We provide the extra detail in the table. We don't provide much information for the non-significant ones. Now, of course, there may be times when we need to provide details about non-significant drug interactions, but those are generally rare, and we'll deal with those on a case-by-case basis. But in general, you should be able to use just the one line. So just quickly, some unique situations I wanted to go over. So the clinical pharmacology section is probably the main place where you'll find information about modeling and simulation approaches to analyzing information. So how do we handle this information? Well, if it's deemed adequate to replace a clinical study, then it should be treated like we treat anything else. So it should be treated like we would treat a drug interaction study, or it should be how we treat a study for a patient characteristic or something like that. So we would give a description of the results, and we'd give information about the design, as needed. Keep in mind that of the on the technical scale, this is the most technical information. So we have to be sparing in how much information we give, and if we need to give a lot of information on the methodology, then we would have to put it in a way that people can understand. So generally, we don't put a lot of information unless we have to about these things. One point I want to make is, if it's deemed adequate, you don't need to always say it's a modeling it's always a POP-PK. So I've seen labels where at least 10 times in the clinical pharmacology section, they say, based on a POP-PK study, this was done. Based on a POP-PK study, that was done. So all the way down. So in general, you don't really need to do that. If you feel compelled to do that and you have most of your data as POP-PK, then just do one line at the top saying the following information is based on a POP-PK model unless otherwise specified. So that way we're not constantly repeating that extraneous information, because you don't need to. In vitro information. So again, the clinical pharmacology section is unique in that we do put a lot of in vitro DDI information, some information about protein binding sometimes, information about metabolic pathways could be also in vitro information. One of the key uses that we have for in vitro information is to establish the absence of a drug interaction. When we think there could be, or we have a positive in vitro study, we generally look to do a clinical study. We have I'll make a plug for our 2 guidances that came out last week, 2 draft guidances on drug interactions. One is a clinical guidance, one is an in vitro guidance. I would suggest everyone read those, and please comment on those, because that's the only way we can make them better, is if we hear comments from you guys. But you'll find in there that really one of the key things in vitro studies does, it tells us when we don't have to do anything else. We don't believe there's a drug interaction. When we believe that there possibly is one, then we generally do something else. We do a study. In some cases, we could do modeling, but generally we do something else. We can put the in vitro information in section 12 if it's clinically important. A lot of times they say it's the protein binding information. Sometimes we can get information on DDI potential and metabolic transporter pathways if we don't have the clinical information. So if we did a study in strong CYP3A4 inhibitors with ketoconazole, we don't need to put in the in vitro section the findings with CYP3A4, because it's already been superseded by a clinical study. So in general, we don't do that. We generally do not put in vitro information in section 7. There are rare cases, and I'm sure people could show me a couple of labels where that information is in there, but that's generally rare because, like I said, the in vitro study alone isn't conclusionary for a drug interaction. We generally do other things to come to a determination about the clinical significance. So generally we find these studies in the clinical pharmacology section. Now this information is also very complex, also very technical, also very hard for a healthcare provider to comprehend when they're looking through this stuff quickly and then they're not basic scientists or not clinical pharmacologists. So what's the bottom line they're looking for? They want to look for, was this studied? Because they may be thinking in their mind, gee, I wonder if this pathway is causing a problem with that pathway. So that's one thing. The one is, was the effect ruled out? That's another thing. And then sometimes when we haven't done studies or we're in a waiting for a study done, they may want to know if there was something positive. And so we could also use alternative display methods to describe this more technical information. And this is just one example. I would challenge folks to try to do some other things with it. I think it's a very complex section that could be improved, and I would encourage folks to try and do that. I gave a talk at the International Society of Systems Antibiotics, the DMPK people, and they were very enthused to see thinking in this direction. But again, they told me I needed to talk to the labeling people, so I'm bringing it to your attention. So just some conclusions. Again, I can't stress enough, essential information, accurate information, directed toward the healthcare provider. We need to be consistent, adhere to guidances and regulations, obviously, and again, the readability and comprehension is key. You could always contact us if you have feedback. Here's our office email address. If there are issues, or generally not issues specific to an application, but if you have, thoughts or questions about labeling, clinical pharmacology labeling in general, or something you want to bring to our attention, you could always email us at that email. For product specific, obviously, go through the project manager and do it that way. Anyway, so thank you, and I'm going to turn it over now to Roya, who's going to tell you industry's perspective on our guidance that we had published last year.
So I hope this works now. Good morning. Again, my name is Roya Babahani. I'm a labeling cluster head from Global Labeling Management at Pfizer. And I'm very excited to be here this morning to talk about this topic. This is the disclaimer you've seen so many times since yesterday, but I don't know why mine is shorter than everybody else. So I was asked to, as Joe mentioned, to give industry perspective on interpretation of the FDA clinical pharmacology guidance, and also to mention some of our internal and external challenges that we face in implementing this guidance, and also in general other guidances. So a lot of my comments apply to labeling in general, not just clinical pharmacology, but there are some certain examples that I'm also going over. So we saw that Joe went through this whole clinical pharmacology guidance and mentioned about the clinical information for clinical pharmacology is mentioned in many sections of the USPI based on the PLR reorganization. So anywhere from the highlights to the clinical pharmacology. And then, again, we saw in his slides of the clinical pharmacology section, section 12 has so many different subsections. So I'm not saying this to repeat everything that is already mentioned, but it's just to say that we don't want to go from one disorganized situation to another, because with clinical pharmacology, we have so many options to where to put the information. And so it is really important to pay attention to that, and we know FDA has done a great job with the PLR, and we know how that has improved the content and where the placement of the information is, and for healthcare providers to be able to get that information. And so we don't want now with the clinical pharmacology having so many options to put things in section 12, to put it under section 7 or 12 on 8 to complicate that. And again, we talked about that FDA has done a great job in providing some guidance documents for the clinical pharmacology. We have the drug interaction guidance, and we have the clinical pharmacology guidance to help sponsors to put the information in the right place to help the flow of the information for healthcare providers to find the information readily. Again, he mentioned the emphasis that is made in these guidances to use the alternative method of the tables and graphs to, in lieu of the text, to make the information clear. Clinical pharmacology content can get complex, so using these tools are helpful. And we talked about, in drug interaction guidance, they encourage the use of forest plot, and Joe again showed the example of that, of the forest plot being used. But my point here is about the challenges. Obviously, from industry point of view, and believe me, Joe, we encourage the labeling teams to use the alternative methods of showing the information, the tables, graphs, and so forth. And we love the forest plot, and we like that to be used more commonly in the label to use for complex clinical pharmacology data. But we also have to keep the end users in mind, the healthcare providers that are not clinical pharmacologists, and they may get overwhelmed by seeing the forest plot and not sure how to read it. And so maybe providing some context around that. We get calls from healthcare providers saying, oh, well, what am I looking at? I mean, I really just want to know what is the percent increase or percent decrease in Cmax and AUC. And so that could just complicate a little bit when they just see this visual display of the information. So we just have to be conscious of the end user, our healthcare providers, what they're going to need at the end. And I know the guidance documents discourage the repeating the information. And definitely I'm not proposing that, and I'm not saying that the same information should be repeated in text or tables. But what I'm saying is that when there need to be an extra context provided to the healthcare providers, that we should do so and explain that what they're looking at in the visual display. So with that, I'm just going to go through some of our challenges that we may see internally. And again, these are not specific to clinical pharmacology. It's just in general for creating the label. And we saw that from some of our other industry speakers yesterday, that most of us in the industry have core labeling or core data sheet that we need to create. And the aim for that is that's our company position, so that usually has to be created first, and then from that we create our local labels, the USPI and SMPC. But when we have a major submission and the timelines are so tight, and we don't have the luxury of that for our core to be developed first, and then from that to create our local labeling, so we have to create all three documents in parallel. So where the challenge or issue comes internally is that in our labeling teams with our subject matter experts, our functional experts from different groups, we are working on three documents that may have different requirements and different needs. So our core data sheet may have a requirement for where the drug interaction should be presented. The same for SMPC might have a different requirement. And then we have to switch and ask our SMEs, okay, now for the US, we have to kind of divide up the drug interaction, the ones with the clinical impact or something that the healthcare needs to do about, that goes in section 7, and everything else is in section 12. So just changing their mindset and switching from one document to another could get challenging. So then some of these issues about creating the table or graphs or things like that could be just specific for US, and that might not be something is encouraged for other health authorities. So anyway, that is some of our internal issues that we have to keep in mind within our labeling teams. This is another example that when we make submissions to health authorities, we normally use the same supportive data to support our information in those sections of the labels. In most cases, obviously in some situations, and when we have this specific conversation with health authorities for specific information that they request, obviously those can be submitted in addition. But the main supportive studies are there, and this is a very simple example just to illustrate what I'm trying to say is that this is for just defining the fasting state for interaction with food, and what gets into the US is at least 1 hour before meal, and what gets into the SMPC is at least 3 hours before meals. And when you go back and look at the supportive data that was sent in to support this statement, the study was at least 3 hours before a meal. So it's kind of hard for us to anticipate or propose the text, not knowing how the health authorities interpret or put the information in the label or ends up at the end. And this is another example with cardiac electrophysiology under section 12.2. In this example, we submitted, there were 2 studies submitted. One was the exposure response analysis, and the other one was the, just the QTC prolongation in the clinical study section, in the clinical study. So what we proposed was a brief summary of both, having the both exposure response analysis and the information from the clinical study. And you can see that's what ended up in the SMPC, having a summary of both studies. But in the US, we only have the data from clinical study. And when I went back to look at the reviewer comment, I'm not sure if we necessarily received rationale or a reason for that during labeling negotiation. But when I went back to look at the reviewer comment was because the QTC prolongation in the clinical setting was, in the clinical study was a mean of, I think, 18 to 20 millisecond, and for the exposure response was around 4. So they thought the clinical study was the more clinically relevant information to put in. However, discussing with our clinical pharmacologists internally, they feel the exposure response information is important, and having that information in the data section of the label, which is section 12, could be important for healthcare providers. So I talked about some of the challenges, obviously, we have to keep in mind with requirements from different health authorities, because as labeling people, global labeling people, we deal with writing labels for different health authorities. But we also get some challenges externally with getting feedbacks from different review divisions of the FDA and in between CDER and SBIA. So when we really feel like, oh, we got it, we know what the FDA is looking for, and then we get something different from another division. And I was really glad to hear that yesterday there are associate directors of labeling in different review divisions. So hopefully that brings that consistency between the different review divisions, but I'm not sure if there's anything in place for consistency between CDER and SBIA. But for a company like ours at Pfizer, we have so many drugs that we basically probably go through all the different review divisions and between CDER and SBIA. So having that consistent way of, for formatting content of labeling, it would greatly help us. The other thing that I've seen recently in some of the back and forth in labeling negotiations, they mention about the Office of New Drug best practice. They refer to that when they make a comment, and I was hoping today that maybe someone that can tell us if there is an Office of New Drug labeling best practice guide that can be shared with the sponsors, because that would be actually very helpful. And then sometimes we don't get consistent rationale from FDA when they make a change, move something, delete something, or if they change the data based on what the analysis that they have done. And I know in my experience, in most cases, every time that we have asked for that information, they would provide it to us. But we may not, that goes to my last point, that we may not have enough time during labeling negotiation to ask for the rationale or why something was deleted or where the data came from. As most of you know, work on labeling, that we sometimes just get a couple hours to get back to the FDA they want our response back maybe in 2 hours. So it's not enough time for us to understand, what the rationale is. So some suggestions for future updates to the guidance. I think it's important to give a little bit more clarity and direction for the cardiac electrophysiology section 12.2. Joe mentioned that right now it's just a very brief statement there about the cardiac electrophysiology. It says if obviously you have pertinent positive and negative, you mentioned that, and they have this standard language if it is pertinent negative, which is good. We also agree with that they're both important. But as also he mentioned that they also mention if you haven't done any QTC, QT prolongation studies, that to omit that header. But we still think that could be important to retain, even though the studies were not done for some of the therapeutic areas, for some of the patient population that you're dealing with, or that they're using co-meds that you want to know that maybe they didn't do any QT prolongation studies. So just maybe keeping that header could be useful. Also, that we have recently seen some new tables being used in, I've seen it in the oncology labels, but I'm sure it might be in other therapeutic areas for interaction, that focuses on clinical impact of the drug interaction and gives guidance on how to prevent it and manage it. This is one example right up from April of this year, and this one, neratinib, from July. That, as you can see, I'm going to go back to this example, that the clinical impact is given, and also how to prevent and manage the interaction. It's very important to put the drug interaction into context and give the clinical impact of it. But what is really important, I feel that this can be used for other things in clinical pharmacology, for section 8, maybe for a specific population, that something, maybe the age or hepatic dysfunction or renal dysfunction impacts clearance or anything, then that you would focus on the clinical impact of that and how you can prevent it or manage it. So I see a lot more use for this type of tables and information to summarize the outcome of the whatever finding you have. And I think the healthcare providers can easily get the information from this kind of table. And as I said, this one was another example, has the clinical impact and how to prevent and manage it. So I think we should, we saw the examples Joe presented, but I think this is another good option to provide that context for clinical relevance of the pharmacokinetic or any clinical pharmacology information. So with that, I think we can take any questions.
And I think you all remember how the game was played from yesterday here in the room. If you have any questions, and look at this, Eric, already up at the microphone there. Good job, Eric. Those of you online, keep the questions coming in the Q&A pod. Both of your mics, so you don't need to worry about using the tabletop. Joe, you've got your mic back on. And obviously this has generated quite the interest. Eric, you were our first one up there. Let's start with microphone number 2, please.
Sure. Just several comments. Fantastic presentations, very clear. A couple clarifications with respect to the drug interactions guidance. So the drug interactions guidance, which contains 3 items, in vitro studies, clinical studies, in vivo studies, and labeling, which was published, draft guidance in 2012. That currently is retired, and currently we have 2, as Joe mentioned, 2 draft guidances out now: the in vitro development and the in vivo clinical drug interaction study development and labeling. So currently we have 2 guidances that just came out in October. And congratulations for that, Joe. A couple other comments about, we do encourage folks to, we agree with you. I agree with that comment about rationale for major proposed changes. So if the FDA has major proposed changes, so the application holder proposes a low dose, mid dose, and high dose, and all of a sudden you see the high dose removed from the labeling, the FDA should provide a rationale for that. Small changes in general, we don't expect that. With respect to early review, we encourage that. That's in the desk reference guide. Obviously, we understand in practice that doesn't always happen. We are thankful for the ADLs, the Associate Directors for Labeling, and the 16 Prescription Drug Review Divisions in terms of coordination to have a consistent response when appropriate, how we implement our regulations, statutory requirements, and our final guidance recommendations. Thank you, and great presentations.
Thank you.
All right, thank you. Let's go over microphone number 1, please.
Yes, thank you. Camille Granville from Bayer Pharmaceutical. Great presentation. I think as a clinical pharmacologist, this is a very good presentation, a lot of input. But my first question is for Joe. I think I have a couple, but I have to limit it. So, and when you're talking about effective half-life, yeah. So teaching clinical pharmacology myself, I think many physicians don't understand what it is effective half-life, yeah? So what they know is the terminal half-life. If I look at one of the table, you also presented as an example. It said elimination half-life. It's not, you presented the data based on the single dose and multiple dose. In the meantime, in the table, it said elimination half-life. So, and I think we have to be consistent in how we translate the message and also, I don't know, from the, you know, the whole clinical pharmacologist to the new, so people doesn't understand what it is, how we calculate an effective half-life. That can be many different ways, so we have to be a little bit, you know, cautious in how we translate those information. The other thing is, I think the pharmacogenomics. In your presentation, this is in the section 12.5. So yesterday we did have a discussion, so whether or not, for example, if we have a polymorphism to D6 to C19, those mutations affect the outcome in terms of the clinical outcomes. So where this information based on the data, where we're going to translate those information in terms of the clinical, whether or not this should be under the section 8 or whether or not this information should be moved under the section 7. So if you want to answer some of those, and also I have comments and for Oya as well. So we can start with you. Yeah.
So with regard to your first question, I agree with you. I think we need to be consistent. I would take it one step further and say the same issue that you're bringing up I have with putting several different AUCs, area under the curve numbers. So sometimes we'll see they'll report AUC, another part of the label, AUC tau, another part of the label, AUC infinity. So yes, I agree with you. I think we need to say, think about, is the distinction important for essential for a healthcare provider to safely prescribe and effectively prescribe this drug? If it is, then maybe we need to explain why we're putting these different numbers. So multiple dosing, therefore we only have AUC tau, that sort of thing. Or if it's just sufficient to put AUC because whether or not it's tau or infinity doesn't really impact what we're trying to do here. So that, you know, again, that's more of a case-by-case kind of a thing, but I agree, we need to be consistent. Same thing with the half-life. I'm not sure we need to necessarily put half-life elimination. I think we could generally put half-life, and it's more of a regulatory issue that we're trying to use the effective half-life. And when we have to use the terminal half-life, we could present that in a way that clearly communicates that we're talking about, you know, it's two different half-lives, and generally those are more the biologics that we're talking about, but we can describe that in labeling. But I agree with you, it should be consistent.
Yeah.
With regard to the genomics, we do have a genomics guidance, so that's the first place I would refer you to. I think it would depend on if the information was used as part of the pivotal trial, whether it would be information would be in 14, or if it was more PK-related information, it might be more in section 12.5. So I think it would depend. And again, that would be sort of a case-by-case kind of thing. But as far as the ClinPharm genomics information and where it should go in 12.5, I would refer you to the genomics labeling guidance.
Okay. Very good. So,
Okay.
and
And
Wow.
your next question, you can follow up back there with the person behind the line. I know we've got a lot of online questions. Let's go to our next online question, please.
The guidance states that the PK and PD values should be reported with a measure of dispersion, such as standard deviation or min-max values. What does the FDA feel like is other acceptable measures, such as percent CV?
I think, again, we need to look at the audience. So as a clinical pharmacologist, as a regulatory officer, terms such as the coefficient of variation are very important to me when I'm reviewing an application. I want to know the variability between the different studies. I need to have that information to make my regulatory decisions. How will a healthcare provider, even though it's technically maybe more correct to use coefficient of variation, how will a healthcare provider use that information for safe, effective prescribing? A lot of times, from what we've heard, we had a advisory committee in 2013 that looked at drug interaction information, at labeling in specific. And what we heard from physicians is they like to know variability. They like to know what the best and worst case scenarios are. They want to know that on average there's a twofold increase in this concentration when we combine these two drugs, but fourfold is also possible. And maybe that's why I'm seeing this person still have this adverse reaction even though I modified the dose or something. So that's important. So we need to really give them useful information that they can use, even though you may be sending us different information because I need that information to make regulatory decisions. So that, I think, goes back to we really need to think about information we need for regulatory decisions, information needed for safe prescribing. Sometimes they're the same, sometimes they may be a little bit different, and we need to adjust the labeling accordingly.
Okay, let's take microphone number 1 here, please.
Yes. Hi, Gina Montero from Eli Lilly, and my question's for Dr. Grillo. The new guidance mentions placing supporting information for immunogenicity in section 12.2 or 12.3. My question is, of course, the clinical impact to ADAs is in section 6 or 5 as appropriate. If the impact, the clinical impact is unknown or there is no impact, what's the expectation for the information to be in section 12?
There's no requirement that the information be in section 12. Most of the time, information will be, as you said, in section 6. The only time you would put information into section 12 is if it is contextual information that supports the conclusions and information in 6. So, for example, if you had more technical PK information that related to inactivation or that kind of thing, you may have that detailed information in 12, but you'll have the ultimate conclusion and the action in section 6. And that's sort of the same for therapeutic drug monitoring as well. So the bulk of the regulatory decision and action information will be generally in section 2. But if you did sort of exposure response or other type studies that support the conclusions that you made in 2, that information you might find in 12.2. So it's really when you have that extra contextual information that you don't want to put in section 6, because having all that volume of information, you could lose the key information with all the context, and you don't really want to do that. So that's kind of why we've been saying sort of separate the information, don't repeat a lot of the information if possible, because really that's another thing we heard when we had the 2013 advisory committee is that healthcare providers are having a hard time finding what's important amongst all that context. So really, that's why we're kind of really trying to hit on, give the essential information and try not to, put stuff in for completeness sake that's really not effective information.
Okay, let's go ahead and take an online question, then we'll come back to the room. Online question, please.
When there is no PK data in patients with renal or hepatic impairment, some labeling omits the section in section 8. However, other labeling says the study has not been conducted in these populations. Is there a rule in this case?
I don't think that there is a formal rule. I think in general, and this was said yesterday, that if there's no information, generally sections can be, most of the sections can be omitted. There are a few that are required sections and subsections. I think what it comes down to is, so one thing is, so for example, you could have in section 12.3 that there's no effect or the information is unknown or whatever, and then really there's no additional context to put into 8. However, I've had feedback from some of the review divisions where they'll say, well, you know, there are issues with renal impairment with this drug, or, you know, they still want to communicate that information in 8 because they feel in this specific situation that it may be important for healthcare providers to have that information there. And in those cases, we've kind of done it. I say in general, you probably could omit the section, but there are cases where they do want to retain those, and those are kind of case-by-case kind of things. Usually, we'll get a rationale from the review division why they specifically think it should be.
Just to add to that, I know we've seen it with the drug interaction as well, that from some review divisions, they omit the section 7 when there are no drug interaction, and then the others that they just add there were no drug interaction studies done. So that's one thing. It can be different, I guess, from different review divisions.
Okay, microphone number 1, please.
Yes, this is for Roya, and also I have something for Joe. And thank you so much for the overview. And I think there's a couple of very good key points. And this is something I was preparing to ask as well. Thank you so much. Regarding the DDI table in the section 7. So I think probably this is one of the most best useful example how to present the data for the clinicians, for the healthcare in terms of DDI prevention. What is the clinical impact? The prevention in terms of the management, and also the question is, okay, should we provide all those you know, different drug that's 3 or 4 inhibitors? So I saw a couple labels from the last, I would say, from last year to now, because there's another BTK just approved, and October 31st, they did the same thing. But I think one of the challenge I feel that after 4 submission myself is really the reviewer preference. Yeah, sometimes they want the information, sometimes not. And I think moving forward, I will really encourage every label to have a section 7, this particular table. This is really very useful for the prescriber with the key information. So that's my suggestion based on your presentation as well. Moving forward, I would like to see every single label the same way. So the second things on the exposure response for the QTC. So this is another question. So for oncology drug where we are unable, if the drug cannot be administered for healthy subject, we cannot do total QTC using moxifloxacin and so on and so on. So we have to do a study in cancer patients. So when I look at your example, so I look at the exposure response, more than 250 subjects. I look at the clinical study, 160. So there's a sample size balance. So the question is, the data is coming from the exposure, yeah, more robust and data set, and also showing that the 90% confidence interval on the upper bound is about 4 milliseconds. So the reviewer said, okay, I need to use the clinical data. In this case, I will ask the FDA as well, if there's a discrepancy between the exposure response and the clinical, should we use additional data, for example, the integrated safety data that has a bigger sample size to back up the information in terms of when we saw there's a discrepancy using the exposure response versus the clinical data?
Fortunately, that's getting more into a review issue and not really a labeling issue. So I would be remiss if I answered for, if you're saying the oncology, for OHOP's reviewers as far as how they interpret data you send in regarding the QT studies they do. I've seen the information that you talk about. Sometimes they don't do thorough QT studies in oncology. Sometimes you actually put more information when they have studies into section 12.2 because it's not a thorough QT study and they feel that they want to put that extra information. Like I say, I mean, I'm a clinical pharmacologist. I could probably answer your question, but I don't feel comfortable answering it for another division.
Eric, did you have something you wanted to add?
Just a comment with respect to the tables in section 7. So currently the guidance gives that's an option of, there are many types of formats. I guess you have to consider that for all human prescription drug and biological products, like dermatological products that may have one interaction. We also have some older drug products where we may not know the clinical impact. So you have to consider when we recommend everything for everything, you could think of some exceptions, but please, the comment period, I believe, is open for the guidance. Please include those comments. Please include when you would want to include it, when you would not. And thank you for your comments.
Thank
And I would
you.
add to what Eric said and say, when you're making comments too, also think about, like he said, if you have 1 or 2 drug interactions, if you have like 5 interactions, or if you have an antiviral with 25 drug interactions, and ideas for how those could be displayed effectively to healthcare providers.
Okay, we've got one more question in the room. I'm going to go ahead and extend our time for 2 minutes. Please.
Hi, Joe. I'm Betsy
Hey.
Scroggs. I'm Associate Director for Labeling for Non-Prescription Drugs, OTC. So I'm here to pick up here what's translational from the prescription side to OTC.
Okay, great.
So one of the things that jumped out at me in one of your slides is when you're just talking about subjective terms we would prefer not to see, especially in the fast, rapid area, which may imply onset. I can see where, of course, if it's defined, it can be useful information they can use. But you would, and I don't want to put you in the hot seat here, but have you really had a chance to think about if we could use those terms, what sorts of data, what are we looking for here if industry could use subjective terms in labeling? How do we define those? Fast, rapid?
Well, so example, if we had fast, parentheses, 20 minutes or 2 minutes, putting some either qualifying or quantifying that term so that it doesn't, so it's not as promotional. I mean, obviously, OPDP would be the best resource to tell you strategies for avoiding using promotional language. But I would say, just from a clinical pharmacology perspective, when we see things like, selective, rapid, near complete, what does near complete mean? Well, 98% in parentheses, right? Those kind of things. So qualifying or quantifying the term is a good way to start. But obviously, the best people to ask that question would be our promotional folks in OPDP. But that would be a general.
We do talk to them, but we have to also rely on the Federal Trade Commission.
Okay, yeah, that you do.
Thanks.
All right, and that's all the time we have for questions.